Hello. In this lecture, I will be discussing about the experiments which was carried out by Frederick Griffith. Griffith established that there was a transforming principle in bacterial genetics through a groundbreaking experiment which he performed in 1928. He postulated that information could somehow be transformed between different strains of bacteria. This was much before the discovery of the fact that DNA was a genetic material. We can now consider that this was the first experiment which was done to give a proof that DNA was a genetic material. Frederick Griffith in 1928 conducted a series of experiments suggesting that bacteria are capable of transferring genetic information through a process known as transformation. We have to remember the fact that Griffith wasn't trying to identify the genetic material, but rather, being a bacteriologist, he was trying to develop a vaccine against pneumonia. So for his experiments, Griffith used Streptococcus pneumoniae bacteria and mice as the host organism. In his experiments, Griffith used two related strains of bacteria known as R strain and S strain. The R strain is also known as the rough strain, and we can see in this diagram the R strain or the rough strain lacks the protective capsule. It is recognized and destroyed by the host immune system. So this makes the rough strain as the benign strain or non-pathogenic strain. So the R strain is non-pathogenic. We can also say that this R strain is non-virulent. So it will not cause pneumonia in mice. While the other strain or the S strain, the other name for the S strain is smooth strain. So I'll write it down over here. The other name is the smooth strain or the S strain. It have a protective polysaccharide capsule which prevents the detection by the host's immune system. So if the mice gets infected by this S strain, the mice will die of pneumonia. So this makes the smooth strain the virulent strain. So S strain is the virulent strain or is the pathogenic strain. Now let us see what the experimental setup was. In the experimental setup, in the first set of experiment, Griffith injected living S strain of bacteria into the mice and the mouse died. As we knew that the S strain is a virulent strain, therefore it killed the mice. In the second set of experiments, living R strain was injected into the mice. R strain is non-virulent or non-pathogenic and we can see the mouse remains healthy. In the next set of experiment, Griffith heat killed the S strain and then he injected the heat killed S strain of bacteria in the mice and he found out the mouse remains healthy. As he heat killed the S strain, he concluded that the S strain has lost its pathogenicity. So the mice remains healthy. In the next experiment, he took the mixture of heat killed S strain and the living R strain and then he injected this mixture into the mice. The interesting results came from this experiment. After injection of the mixture, the mice died, indicating that some sort of information has been passed from the dead S strain to the live R strain. Further, blood sampling of the dead mice showed that live S strain bacteria was present in the blood of the dead mice. So what can be concluded from this experiment? Griffith concluded that the R strain of bacteria must have taken up what he called transforming principle from the heat-killed S bacteria, which allowed them to transform into S strain bacteria and became virulent. So what Griffith concluded, that this was the heat-killed S strain. So S strain was heat-killed. He concluded that the heat-killed H strain, from this heat-killed H strain, and this was the live R strain. What Griffith concluded, that something from this heat-killed H strain has transformed this R strain bacteria into living H strain bacteria. So this is H strain bacteria which is living. So something must have transformed the R strain into living S strain, and that something must have come from this heat-killed S strain. Now this was the conclusion of Griffith, and he said that transforming principle has the capability to transform one strain of bacteria into the other. We should now remember that Griffith did not conclude about the nature of the transforming principle, but follow-up experiments performed by Avery, MacLeod and McCarthy and by Hershey and Chase established that DNA was the mechanism of transferal of this genetic information between the two bacteria. Hope this was helpful. Thank you.